Hey everybody, today we are going to discuss plasmid DNA and gene modification. But, first we must ask ourselves, what is a plasmid? And plasmids are found in prokaryotes. Found in prokaryotes. And basically they are circular double-stranded pieces of DNA, so just kind of like our DNA, but circular. They are comprised of the four main base pairs: adenine, thymine, cytosine, and guanine. So they are circular, double-stranded pieces of DNA comprised of adenine, thymine, guanine, and cytosine. So, now a lot of people want to know why we use these plasmids, these circular double-stranded pieces of DNA in genetic modification. Well, like I said before, they are used in prokaryotes, which are essentially bacteria. And there are three different parts of the plasmid, right? So there's the origin of replication, the multiple cloning site, and the antibiotic resistant cassette. The origin of replication is the portion on the plasmid where DNA replication is initiated. The antibiotic resistant cassette is the gene on the plasmid that codes for proteins that can inhibit antibiotics from killing the bacteria. And then the multiple cloning site is the place where the gene of interest is inserted, and it contains restriction sites, which are here in these purple boxes, that have specific enzymes that can cut at these specific sites. And the real big portion that is used in genetic modification is this site, is the multiple cloning site, or the MCS as we call it. So using this model of DNA that I have constructed from origamindna.com, we have this. We have our multiple cloning site, and what we want to look at here is we have some outside DNA that's outside the multiple cloning site, and then we have this piece of DNA that's inside the multiple cloning site. This is just usually DNA that codes for different proteins or that contains different genes that create compounds or certain things, whatever the bacteria wants to use that specific site for. So what we can do is we can digest the plasmid using restriction enzymes. Now here's the thing about restriction enzymes. These are going to be represented by my scissors here. Restriction enzymes are used to cut the DNA out of the plasmid, and one thing about restriction enzymes that is really neat and that what we can use is they cut at specific sites on the DNA. Now, only in these restriction sites, as we call them, are comprised of approximately 6 nucleotides that are generally that are very specific for a restriction enzyme. So basically, a restriction enzyme can only cut at that specific restriction site. So let's just say we have restriction enzyme A and we have restriction site A. Restriction enzyme A will cut at that specific restriction site. However, restriction enzyme A cannot cut at restriction enzyme B or restriction site B. And that's just because of the specificity of the 6 nucleotides in that site. So basically what we can do is we have a restriction enzyme, and it will cleave at specific sites. So it'll cleave here, and then let's just say there's a different site, and we're using that different enzyme that is specifically for that site, and it could cleave here. Now there are multiple, and our DNA that we've cut out will go away. So now we have our digested plasmid, and just picture the DNA just as kind of like a circular plasmid. And we could tell that our digestion worked because restriction enzymes are, the plasmid DNA is going to be missing a big portion of the DNA. So we run gel electrophoresis, which is a method of seeing how big our DNA is. Obviously, since we cut out a piece of DNA, it's going to be smaller. So then, well, now what do we got? We got this multiple cloning site with nothing in it. It's digested. So now what do? Well, we want to put in our colorful gene of interest. Now the gene of interest could be any specific thing that we want, whatever protein that we need coded for, or if we want to provide a specific function, we would use, it just kind of depends on that situation. And we want to basically insert our DNA into the multiple cloning site. So how do we do that? We use DNA ligase, represented by tape, and the DNA ligase is an enzyme that allows for the binding of DNA at the primers on the gene of interest. The gene of interest has these little primers on the end here, and basically what will happen is DNA ligase will bind the primers to the restriction sites, and these primers have base pairs that are complementary to the restriction sites where we made the cut. So our DNA ligase is going to come in here, it's going to tape or splice together the gene of interest, and then it's also going to come here and splice in the gene of interest over here at a different restriction site. We use DNA ligase to splice in our gene of interest. Now, one thing good about DNA ligase is it's not specific for the restriction sites, so DNA ligase could bind the primers on the gene of interest to the corresponding restriction site DNA, just with regular enzymatic activity for that. And then another thing that's really neat about, so once our DNA has been inserted, we will be able to, coming back to our plasmid here, we will be able to insert the plasmid DNA, which is this, with our newly set gene of interest, which is all this, and put it back into bacteria, the bacterial cell. Once it's in the bacterial cell, we'll give it a couple days to replicate, just through basic DNA replication, which is initiated at the origin of replication. And then we will select the bacteria that have been successfully transferred with this DNA by using an antibiotic that is corresponding to the antibiotic that the resistant cassette that produces an antibiotic inhibitor for a specific antibiotic. So one common thing that we use is kanamycin to kill the bacteria. And let's just say this bacteria is resistant to kanamycin. Well, our gene expression has been turned on by the insertion of the multiple cloning site. So whatever has not been inserted is going to die because the antibiotic gene has not been turned on by gene expression. Whereas the ones that have our gene of interest in our multiple cloning site, those are going to survive basically because our bacteria are not producing the antibiotics antibiotic inhibitors that are needed for survival. So y'all are probably wondering, well, Tanner, why do we care about this? And what is this main use of this plasmid DNA and everything? So one thing I want to say is, once we have our beautiful piece of DNA here, where we have inserted our gene of interest into, we're going to take this segment right here and put it into the multiple cloning site. So this part is going to equivalent to here. You're going to have your origin of replication, and you're going to have your antibiotic-resistant cassette. This whole entire plasmid will be inserted into agrobacterium. Agrobacterium, they normally will bind to pollen on the plant Arabidopsis, or just on any plant. And one thing that they like to do, naturally with their own DNA, is they take their DNA, they transfer it over into the plant's genome. When the plant has the DNA in their plant genome, what they do is they will take the take the DNA and they make opines. Now, opines are a type of nutritious compound that cannot be produced by agrobacterium alone. They have to be produced by plants containing that agrobacterium DNA. Hence, why agrobacterium put the DNA into the plant. So, what we can do is usually this, remember I was talking about the chunk of just junk DNA being in here? That's what we remove, is that opine production gene out of the agrobacterium, and we put our gene of interest in here. Now, our gene of interest can be used in multiple ways. It could be used for repressor genes, inhibitors, activators, enzyme cofactors, just any type of hormone or protein producing or protein compound that will that we are wanting to use in our research. And one thing good about having our new modified DNA is we can insert it into the agrobacterium, and the agrobacterium are going to use that DNA even though they don't have that gene to produce opines. It doesn't really matter. That's still part of their biological process, is to bind to pollen, transfer the DNA in hopes for opine production. So we've essentially kind of tricked the agrobacterium into producing whatever we want to produce in our gene of interest. And I want to thank you guys for taking the time to watch the video. I hope you learned a lot about plasmid DNA and gene modification. One thing I want you guys to, I'm going to leave you guys with the thought is, how could you guys use this in research fields if you guys were to go into research, whether it be in medical and plant or just any other type of research that involves DNA modification and the use of bacterial transformations. I hope you all have a good day, and thank you.